Parkinson's disease is a neurological disorder that affects 1 in 37 people alive today. It is mainly caused by the loss of dopamine-related neurons in the brain, which results in symptoms such as tremors and the loss of motor function. Although it is not known exactly how dopamine-related neurons die, it is believed that oxidative stress plays a key role in this, so research into this area is essential. Research in neuroscience is usually conducted in C. elegans, since their neural maps are seen as somewhat simple and are fully catalogued. Elements of dopamine neurotransmission in C. elegans are also highly conserved in mammals, which makes them ideal for the study of Parkinson's disease. By looking at the effects of various drugs that cause oxidative stress, such as Paraquat, hydrogen peroxide, and 6-hydroxydopamine, it is possible to see the various mechanisms by which cell death and protection can occur. A recent study carried out at the University of Dundee did exactly that. The study discovered that the gene TTR33 inhibits neuron degradation induced by exposure to 6-hydroxydopamine.
Neuroprotective genes were identified through the use of VTIS1 C. elegans strain. This strain allows for the selective visualization of dopaminergic neurons through the expression of GFP, green fluorescent protein. These animals were then mutagenized with ethyl methanol sulfate throughout various generations and exposed to levels of 6-OHDA that don't normally cause neurodegeneration of the dopaminergic neurons in wild-type C. elegans. Any mutant that did exhibit this type of neurodegeneration was isolated and genomically screened to find 2 specific point mutations in the transthyretin family domain TTR33 that affected the TTR33 protein. This resulted in a loss of function of this protein, increasing the 6-OHDA sensitivity. Researchers discovered that TTR33 played an important role in oxidative stress tolerance during the L1 and L2 larval stages in C. elegans. Exposure to 6-hydroxydopamine, that is 6-OHDA, after the L3 larval stages showed little to no effect. Deletion of the DAP1 gene prevented 6-OHDA uptake and therefore also prevented the dopaminergic neurodegeneration.
In order to see the overall effects of oxidative stress on the whole organism, C. elegans were put in a shaker for 1 hour with H2O2 and Paraquat. It was shown that there was an overall insensitivity to oxidative stress, and this showed that TTR33 played a very important role as a scavenger of free radicals in C. elegans.
So essentially we don't know the answer. We simply don't know. It's, again, just when you do science, well, there's 2 ways. You either do nothing, and then of course nothing comes out, or you make a decision, and the decision is essentially like jumping into the cold water and see what happens. And of course that's what we had to do in our project, since it was something entirely new. And for that reason, we of course had to accept initial limitations, and one of the limitations of the system certainly is that actually we typically assay neurodegeneration in relatively young animals, which is also a very convenient way of actually doing the assay. Sort of technically it makes it easier. But yes, that's a concern, but yes, we simply have to start somehow.